Good afternoon. My name's Aidan Scully, student number 176825. This is task 3 of Biology Unit 4, Infection and Immunisation assignment. This assignment will mainly be talking about the human body's immune response. When a pathogen tries to enter the body tissue or bloodstream, whether it be a bacteria, a virus, even a parasite or fungi, it doesn't matter at this point what type of pathogen it will be, because the first non-specific line of defense that will try to deny the pathogen access to the body will be the physical barriers. Now, a normal person's body always strives to maintain homeostasis. Homeostasis is a natural state of balance of all of its organs, nervous, respiratory and circulatory systems. This homeostasis can be disturbed by harmful pathogens. The first line of defense against pathogens is the immune system's natural barriers. These mechanical barriers include the skin, which is composed mainly of keratin. Keratin is a tough protein which is hard for most pathogens to penetrate. Also, on the surface of the skin, there's sweat, hairs, and the natural pH level of the skin, as well as oils produced by the sebaceous glands. All of these help to limit or kill microbes. Hairs in the nose and ears also form a barrier in the form of small cilia, which catch pathogens and foreign debris, stopping them from going further into the ears and nose. Mucous membranes of the digestive, respiratory, urinary and reproductive tracts, as well as the ears, nose and throat, are open to the exterior. This makes them vulnerable to invasion. They are protected by mucous membranes that physically ensnare microbes. Once caught, these microbes are either flushed out via the urinary tract or flushed out by saliva and tears. They can even be swallowed and sent towards the stomach, which they will then be killed by the stomach acids. Mucus, tears and saliva also contain lysozyme. Lysozyme is an enzyme which destroys bacteria by dissolving their cell walls. Now, if a pathogen can bypass all of these first line of defense barriers, perhaps through a cut or even a broken layer of skin, then the second line of non-specific immune response is activated. This second line is considered non-specific due to the fact that it cannot yet start targeting a specific type of pathogen or organism, but rather it is a defense and response which protects against pathogens as a whole. This defense system is also known as the innate defense system. Internal innate defenses can include fever, inflammation, and the release of chemical signals to defensive cells. Damaged cells can send chemical signals that attract phagocytes. Phagocytes derives from the name, which literally means to eat. They come in a few different varieties. The most abundant are the neutrophil variety of white blood cell. They devour pathogens and then virtually self-destruct themselves. The remaining parts of the cell forms pus. The other type are the macrophages, which are derived from monocyte white blood cells. Monocyte white blood cells which have moved out of the bloodstream to occupy tissues. There are two types of these monocytes: the free type that patrols tissues looking for pathogens, and then there's the fixed type that attach to fibres within specific organs and devours anything foreign that passes by. When a macrophage is close enough to a pathogen, it snares it by extending its cytoplasmic extensions. It reels it in, engulfs it, and essentially digests it. Unlike neutrophils, it can do this many times. Natural killer cells, or NK cells, also patrol the blood and lymph looking for pathogens. They can even kill your own body's cells if they are infected with viruses or have become cancerous. But how can the innate immune system detect invaders? The body wants to contain the spread of pathogens, clean up the dead cell mess, and get healing as fast as possible. So it sparks up your inflammatory response. It is basically an internal fire alarm, except it utilizes chemicals instead of sirens to get the message out for help. The body or infected area experiences redness, swelling, heat, and pain. Specialized mast cells in your connective tissues send out histamine molecules. Histamine causes vasodilation, which creates redness and heat near the widened capillary surrounding tissues. The increase in heat raises up the cell's metabolic rate so that they can repair themselves faster. Other chemical signals cause the release of proteins, which can also cause inflammation, clot blood, and form scabs, while the lymphatic system sucks up and filters that extra fluid before introducing it back into the bloodstream. Local inflammation attracts phagocytes and lymphocytes, which flow freely to kill pathogens and clean up dead wreckage, i.e. from neutrophils, which, as they are the first responders, have already started to die. The injured skin cells release chemicals that begin leukocytosis, the release of neutrophils from the bone marrow where they are made into the bloodstream, to attract the new neutrophils to the infected area. Endothelial cells in the capillaries send out chemicals that act like homing devices. The neutrophils cling to the sides of the capillaries and then squeeze through in order to find more pathogens to kill. Sometimes neutrophils and macrophages can get overrun with pathogens. When this happens, they let loose pyrogen chemicals which signal to your body to raise its thermostat and cause a fever. This increase in body temperature again increases the metabolism of your cells so that they can heal faster. The chemicals also tell the liver and the spleen to hold on to all of their iron. Also, it tells the liver and spleen to hold on to all of its zinc so that those elements can't contribute to any more bacterial growth. Now, if none of this helps and the infected site is overwhelmed with pathogens, it's time to activate the third line specific immune defense, otherwise known as the adaptive immune response. Due to its ability to target and memorize specific pathogens and learn how to quickly kill and overcome them. The adaptive immune system needs to be introduced to a specific form of pathogen before it will attack. Phagocytes become professional antigen presenting cells after breaking up pathogens and displaying its engrooved proteins on its outer membrane. The adaptive immune system never forgets a pathogen once it has come into contact with it. Your adaptive system can also fight pathogens throughout any part of your whole body. They can do this simultaneously at once. The main trigger for the adaptive immune system are a pathogen's antigens, which are large signaling molecules that are not normally found in the human body. They act as huge flags to the adaptive immune system. With any luck, a pathogen will bump into a B lymphocyte cell, which is part of your humoral response. B cells are created and matured in the bone marrow. Once matured, they can determine friend from foe via immunocompetence. Immunocompetence is the ability to recognize and bind to a particular antigen. B cells also have the ability of self-tolerance. They know how not to attack your body's own cells. Once fully matured, a B cell displays at least 10,000 special protein receptors on its surface. These are its membrane-bound antibodies. Each B cell has its own set of antibodies, which only fit a particular type of antigen. Now, your immune system tries to win the battle by using overwhelming odds. Imagine 2 billion keys, and each one fits a different pathogen's antigen. B cells colonize and roam around your blood and lymph node. They won't be truly activated until they come across their perfect antigen match. When a B cell finally finds an antigen match for its antibodies, it binds to it. The B cell is now activated and starts to clone itself into an army of B cells with the exact same antibodies it started with. Most of the clones become active fighters or effector cells, but a certain proportion become memory cells which preserve the genetic code for the successful matching antibody. Now, if and when the same antigen returns, there will be a prepared secondary immune response. This secondary immune response can kill the pathogen quickly before you even begin to feel its effects, or even possibly before you become ill at all. Effector or plasma cells can mass produce antibodies at around 2,000 antibodies per second. Now they can do this for 4 or 5 days until they finally die. These antibodies do the same job as the B cell surface antibodies. They are now just free floating. These antibodies bind to all the antigens they can find and mark them for death. Antibodies can cause neutralization. Neutralization of a pathogen is whereby enough of the antibodies are attached to the pathogen so that they can physically block the antigen sites, and that antigen can no longer connect to and harm any of your cells or tissues. The antibodies that are free floating can also attach to more than one antigen at a time. This binds antigens together in a process called agglutination. The antibodies can also send chemical signals to phagocytes so that they can come and kill and digest these pathogens that all link together. Now, if the humoral immune response fails to stop infection, it now becomes the last and final fight with our cellular immune response. T lymphocytes or T cells go after body cells that have been hijacked by pathogens or become cancerous. T cells can also cause inflammation and activate macrophages and call on other T cells. T cells are the main regulators. They regulate much of the immune response system. Dendritic cells have proteins on its surface which can bind to fragments of miscegenous antigens. Miscegenous antigens are parts of a broken up pathogen. They do this to get the attention of other cells. This is useful as the T cells cannot detect whole antigens. They can only detect these fragments. T cells come in the form of helper cells, cytotoxic cells, memory cells, suppressor cells, and natural killer cells. Helper T cells can't kill, but they can activate and signal different cells that can kill. The cytotoxic T cells actually do all of the killing. But helper T cells, much like B cells, only have receptors that will bind to a specific antigen's fragments. Just like the helper B cells, once activated, the helper T cell starts cloning itself and also making memory T cells. Helper T cells then release chemicals called cytokines. Cytokines are chemicals which cause other C cells to clone themselves. This process causes those T cells to release their own cytokines, and so on and so forth, so that the spread of T clone production and cloning happens throughout the body. This enables more T cells with the correct antibodies available to track down and kill the invading pathogens.